Thank you, Gillian. I'll pass it on to you.
Good morning, everyone. I'm Jill Slater and I'm your trainer for this morning. Can I just make a point? It's probably easier if you put any questions in chat box and I will look out for them. And that's how we normally do it when we've got a lot of people at a presentation, because if everybody tries to speak, it can get a bit chaotic. So that's the usual way that we raise questions on this type of presentation. This is the agenda for this morning. This session introduces important aspects of the course, focusing on the specification, planning to teach the course, and there's also an overview of assessment and the support available from Pearson. This part of the training is planned to run for 2 hours. And I've suddenly got a problem because my screen has frozen. That's it. So this is the aims and objectives of this morning. We're going to look at how the qualifications are devised, review the content of the qualification, explore how to plan the course and to plan lessons to deliver the course, understand assessment of the qualifications and how you can prepare your students, and then we will identify the support available from Pearson and network and share ideas with other teachers. Pearson is the world's leading learning company and the UK's largest awarding organisation. Our international heritage stretches back over 150 years. And together we partner with schools, universities and employers, offering globally recognised qualifications to over 3.5 million students a year. We're going to start off by having a look at the specification. This is the main document that you need to teach the course, and you will find it invaluable in your teaching. The latest version of the specification is available to download from the Pearson website, and it's also available to download from the resources that you've been sent for this training. If you look at the contents page, that will take you to the key parts of what you need when you're planning for teaching and assessment. For instance, the biology content is listed between page 12 and page 42. This is the biological knowledge that you need to teach your students and that your students need to learn. And you need to cover the whole of this part of the specification. The questions on the assessments are all compulsory. There's no choice. The assessment information will be found on page 43 to 46. And this section outlines the design of the assessment, including details of time and marks for each examination and details of how each unit assessment contributes to the final aggregated grade. A further aspect that you need to make yourself and your students aware of is the mathematical skills in Appendix 6. This appendix lists the mathematical skills that can be assessed during the lifetime of this qualification. Each skill is exemplified in the appendix. However, the skills can be assessed in any appropriate biological context. A further aspect that you need to make yourselves aware of is the taxonomy, sometimes referred to as command words, which you will find in Appendix 7. This appendix outlines the key vocabulary used in the specification, but more importantly, in the examination pages. It's important that your students are familiar with this vocabulary. Another resource available to you are the SAMs, Sample Assessment Materials. These were produced when the new qualification was developed, and they are examples of questions and mark schemes, and they can be considered as templates for all subsequent external assessments used during the life of the qualification. There is a stamp for each unit examination, and they can be found on the Pearson website. They're available to download. And now we're going to look at how the content is assessed. And it is a cyclical process, but it begins with the production of a question paper. Each of the examination papers used in an external assessment is written by the principal examiner. The paper is then reviewed and revised by a committee of experienced senior examiners. At the same time, a provisional mark scheme is produced. Students then sit the examination paper, and their answers are scanned into the marking software. All examiners contracted to mark the paper will then prepare for marking by looking at student answers. Each examiner provides feedback to the principal examiner. This feedback is then used to revise the mark scheme to take account of student responses to the questions. At this point, a final mark scheme is produced. The final mark scheme is then used to standardise the marking of all examiners. This process ensures that each examiner marks consistently. Throughout the marking process, the accuracy of each examiner is closely monitored. This is achieved by two methods. First, a set of student responses with an agreed mark are given to examiners to mark in amongst their live marking. Secondly, senior examiners select samples of each examiner's marking and check that the marking is of a sufficiently high standard. Once all the marking is complete, the process of awarding is undertaken. During the awarding process, candidate performance for the cohort being assessed is compared with previous cohorts and with the requirements for the award of a grade. To do this, the awarding committee balances a range of evidence. We will consider how marks are turned into grades in more details later. Following awarding, the principal examiner for a paper will reflect on the performance of the paper. In particular, the principal examiner will look at questions that did not work as expected and use this to help refine the writing of the next paper. Now I want to take a closer look at the specification. To start with, we will consider the specification points We will consider the biological knowledge and understanding in the specification. We will not be looking at every specification point. There are 170 different points in the specification. Rather, we will consider how the specification is organised and how to interpret the specification. The biology content is found in pages 12 to 42. The content is split into 6 units. 4 units, 1, 2, 4, and 5, detail biological knowledge and understanding that can be assessed in the examinations for these units. 2 units, 3 and 6, describe how the practical skills candidates need to develop during the course will be examined. It is envisaged that practical skills developed in units 1 and 2 will be assessed in the unit 3 examination, and practical skills developed in all 4 units, 1, 2, 4, and 5, will be assessed in the unit 6 examination. So we have got In the International AS, we've got 2 units, molecules, diet, transport and health, which is divided up, and cells, development, biodiversity and conservation. And then in the second part of the course, for the A level, IA2, we've got a further 2 units, energy, environment, microbiology and immunity, and respiration, internal environment, coordination and gene expression. Units 1, 2, 4 and 5 follow the same format. The topics covered in these units are very much the foundation. The IS is units 1, 2 and 3. The topics covered in these units are very much the foundations or building blocks required by the students undertaking an A level in biology. So these are the basics that you would teach first if you were embarking on A level biology. If we go back to the specification, pages 14 to 24 detail biological knowledge and understanding that will be assessed in examinations for units 1 and 2. Each unit is split into 2 topics. Topics comprise specification points that relate to a broad theme. So, for example, if we go back again, the 2 themes in topic 1 are molecules, transport and health, and membranes, proteins, DNA and gene expression. And then in topic 2, we've got cell structure, reproduction and development as one topic, and plant structure and function, biodiversity and conservation as the second topic. Pages 20 to 25 to 27 of the specification describe the practical skills that will be assessed in the examination for Unit 3. It is important to recognise that the practicals used to develop practical skills assessed in the Unit 3 examination are described in the specification content for Units 1 and 2. The IA2 follows a similar pattern to the IAS. Units 4 and 5 detail biological knowledge and understanding that will be assessed in examinations for these units. Unit 6 details practical skills that will be assessed in the examination for this unit. Again, these practical skills can be developed using the practicals included in the specification content for units 1, 2, 4 and 5. The topics covered in the IA2 are more demanding than those in the IAS, bringing together basic concepts and preparing students for the rigours of higher education. And there's a question about the codes for the exam papers. Yes, they are WBI 11 up to WBI 16. And the mark schemes will match the specification. Now, as we go on, we're now going to have a look Sorry, I'm just trying to keep up with any questions in the chat box as well. We're going to have a look at how the specification can be interpreted. So if we look at an example, this is an example of part of the specification. Now, as you look at a unit, this is taking just a part of it. But as you look at the unit, you'll get a short introduction, which gives the philosophy or biological content of the unit. Opportunities for developing practical and mathematical skills are also identified for each unit in each part of it. The introduction is followed by a brief description of the assessment for the unit. The content for the unit, as we have been talking about, is split into two broad topics, and each topic is split into a number of specification points. There are around 20 individual points for each topic. So you do need to make sure that you've covered all of the specification points. The exam can be on any one of the points. The aim of the exam is to cover as many specification points as possible. So these are two specification points taken from topic 1. I'll give you a couple of minutes to read them. Now, this tells you what your candidates need to know. If it's not in here, it's not something that they need to know. So they do need to know about the role of thromboplastin, the roles of thromboplastin, prothrombin, and fibrinogen in the clotting cascade, because that is specified in the specification. They don't need to be taught about regulatory factors or any details about the different mechanisms triggering the release of thromboplastin. For 120, the candidates need to know the benefits of hypertensives. So in order to know the benefits, they would need to know what they do. For instance, reduce blood pressure. They're not expected to give the name of a hypertensive or describe the mechanism of action of a hypertensive. The specification points identify what needs to be taught. So candidates are only expected to know about the biological terms that are listed in the specification. And the same applies to the names of enzymes, biological molecules, and biochemical details, such as types of bonds. So anything that is in the specification, they should be expected to be able to answer a recall question on any aspect. However, teachers need to use their professional judgment when interpreting the specification points, particularly around biological processes. And candidates also need to be aware that they may be asked a question that's set in a context. So if they look at a question and think they do not know, they've not been taught this, then in all probability, all the information they need to answer the question is given in the context of the question, and they have to apply the things they have been taught to that particular question. If we move on, this is a specification point from topic 6. Understand the differences between the roles of B cells, B memory and B effector cells, and T cells, T helper, T killer, and T memory cells in the host's immune response. The idea behind this specification point is that candidates should understand the different roles of B cells and T cells in an immune response. If we just look at the B effector and T killer cells, so we select one type of B cell and one type of T cell, it's reasonable to expect that candidates will be taught that plasma cells are B effector cells, and that each plasma cell or clone produces antibodies that recognize a specific antigen. They should understand that these antibodies are released into the bloodstream and then bind to antigens in the blood, e.g. on the surface of bacteria or viruses, or on the surface of cells lining the blood vessels. Equally, they should be taught that T killer cells have receptors that recognize specific antigens on the surface of the cells, e.g. viral proteins on the surface of virus-infected cells. They should understand that killer cells bind to the antigens on the surface of these cells and lyse them. They would not need to know about different antibody classes or the role of MHC antigens or the roles of other effector cells such as mast cells or natural killer cells. Now, you should have with you, you should have downloaded the specification. It was in the resources pack that was sent to you. However, I can give it you here. Understand the role of glycolysis in aerobic and anaerobic respiration, including the phosphorylation of hexoses, the production of ATP by substrate level phosphorylation, reduced coenzyme, pyruvate, and lactate. It tells you that the details of intermediate stages and compounds are not required. So, which statements about, can you write down or put in the chat box the statements about glycolysis that you think your students should be taught, and then we will go on to have a look at some of the statements that might have been taught and some statements which do not need to be taught. So put into the chat box what statements about glycolysis you think your students should be taught. Right, I've just had a look in the chat box. There's a number of things I'll come back to. Firstly, this is not an exam question. This is thinking about how you would teach the topic. So that's why putting which of these statements should be taught is fine. You wouldn't find an exam question that said which of these statements. You're quite right, it would be more specific and it would use the detail, it would use one of the command words. When we go back to, sorry, I missed, there were some questions about antibodies and B cells and T cells. I'm not familiar with the Anne Fuller textbook. However, Pearson does, when we get onto Pearson's support and Pearson's resources at the end, they do produce textbooks that are specifically linked to this course as it changed in 2017. And that should get that. I've not taught IAL. I have taught the English A level, but I have used Pearson textbooks and I have found that they give a good indication of the depth of the knowledge, because I appreciate it's not always easy to interpret exactly how much detail you want to go into. The immune system is a system that students do find hard. Having marked plenty of papers with questions about the immune system, I appreciate that they do find it difficult. However, it is to some extent just a question of learning the facts about the different types of cells. It is hard to remember, they just have to find ways of doing it and then applying it to the question that's asked, because we'll come on to assessment, but students do tend to lose marks because they don't actually answer the specific question that's asked. If we go back to looking at respiration, we've got lots of ideas about what you should think about teaching when you're teaching about respiration. If we move on to look at the statement 7.2, which is this statement is specifically about glycolysis. The topic goes on to talk about Krebs cycle and the electron transport chain, but this specific statement is about glycolysis, and it specifies phosphorylation of hexose, the production of ATP, reduced coenzyme, pyruvate, and lactate. So you would expect your students to know that for it, or you would expect to teach them that for each molecule of glucose undergoing glycolysis, 2 molecules of pyruvate are produced. You would expect them to know that glycolysis starts with glucose. They should know that NAD is reduced in the production of pyruvate and then oxidized in the conversion of pyruvate to lactate. You would go on, when you were teaching about Krebs cycle and the electron transport chain, to talk about the oxidation of NAD later on, but not as part of glycolysis. You would expect them to know that ATP is produced during glycolysis by substrate level phosphorylation. And you would expect them to know that for every 2 molecules of ATP used to phosphorylate hexoses, 4 ATP are produced. So there's a net production in glycolysis, in the glycolysis part of respiration, that produces 2 ATP. So that's what you would expect them to know. You don't need them to know that fructose is phosphorylated to fructose 6-phosphate, nor do you nor do they need to know that in glycolysis, the enzyme adding the second phosphate is called phosphofructokinase. These are not part of the biochemical detail that is in the specification. So candidates are not required to know this. Phosphofructokinase is an enzyme that is not named in the specification, so candidates do not need to know this. But as I said previously, even though candidates are not expected to know something, this does not mean that they will never come across them. They may be given appropriate information in the examination that asks them to interpret some data to explain the role of a named enzyme in a biochemical process such as glycolysis. And in this situation, they would be applying their understanding of glycolysis and of the way enzymes work. So you need to encourage your students to make sure that they always read exam questions carefully. And if they look at a question and think, I haven't been taught this, to read it again very carefully and decide what they have been taught that will enable them to answer that question. So we're now going to look at how the practical skills units are organised. They are organised a little differently because the page on the practical skills will have an introductory paragraph, a list of practical skills that will be assessed and that need to be developed through teaching and learning, and they're listed as a set of bullet points. More details of the assessment are then provided under 3 headings: planning, implementation and measurement, and either processing results for Unit 3 or analysis for Unit 6. Within the specification, there are a number of core practicals and a number of recommended practicals. Core practicals can be considered as essential in developing students' understanding of the subject and in preparing them for the practical skills assessments in Unit 3 and 6. So students need to be really familiar with the core practicals. The recommended practicals are not essential, but they will support student learning if it is possible to undertake them. And practicals shouldn't be seen merely as a tick box add-on exercise. It's very important that they are used to teach students the required practical skills to enable them to perform well in Unit 3 and Unit 6 examinations. And students, where at all possible, should carry out certainly all the core practicals, and where possible, at least some of the recommended practicals. Practical skills are developed by actually carrying out the specification. Actually carrying out the practicals. So there's a number of questions assessing practical skills. The growth of pollen tubes is not a core practical, but it is a recommended practical. And this again will help develop the skills that are needed. So One of the recommended practicals is to investigate factors affecting the growth of pollen tubes. Can you quickly type into the chat box what sort of factors you think you might investigate if you are carrying out this practical with your students? And we're getting some good examples of what you might change or investigate: temperature, concentration of minerals needed for growth, the age and type of pollen, the pH that it's grown in, concentration of glucose. That also is important. The length of the pollen tube is what you will be measuring rather than investigating, probably. But effective temperature, humidity, glucose concentration. Lots of ideas of the sort of thing that you could investigate. Now, in the Unit 3 examination, candidates will often be asked to devise a procedure to investigate something. These questions carry a relatively high mark tariff, and many students find them challenging. This type of question will normally be set in a particular context, and candidates will need to use both information from the question and apply their knowledge and understanding gained from practical work to gain full marks. This type of devised question is unlikely to be just a complete reiteration of the core practical. It requires students to apply their knowledge to the context of the question. Now, if we have a look at a question from a past paper that's based on a practical. So this is again about pollen tubes. It shows the results of an investigation into the effect of boric acid concentration in a sucrose solution on the growth of pollen tubes. Many of you, in suggesting what you could investigate, referred to glucose concentration. That is fine, because glucose is used in respiration, which is what it's required for. Because sucrose is synthesized by plants, quite often pollen tubes are grown in sucrose solution, but your students wouldn't be penalized for using glucose, because that's absolutely fine. So this question asks candidates to devise a procedure to investigate the effect of different concentrations of sucrose on the rate of growth of pollen tubes using this sucrose solution. So they started off with 500 grams per decimeter cubed. They were investigating the effect of different concentrations. Before we think about how they might, what they might actually write, we need to think about what information they're taking from the context and what do you think they are taking from the knowledge they already have. So can you all put in the chat box the information you think the candidates need to use from the information provided in the question? So if you start by putting in the chat box what you think they needed from the context of the question. We have some comments in the chat box. Thank you. Yes, we're getting some good ideas here. They're given a starting solution of sucrose solution, so they should base their concentrations on that starting solution. Boric acid is not something that they will have used probably doing their own experiments, so they need to take information from that and decide on the concentration of boric acid they're going to use and maintain it as a constant throughout the experiment, and they need to think about time, how they're going to time it. And then when they've got that information, they should then use information from practicals they have carried out, such as knowing that there should be a minimum of 5 different concentrations of sucrose solution, that they will use a microscope and graticule to measure the pollen tube length at specified time intervals, and identify other factors such as temperature that need to be controlled. So this summarizes that. Your 5 concentrations is from practicals they've carried out, starting with 500 grams per decimeter cubed of sucrose solution is from the context. Boric acid is from the context. Microscope and graticule is from practical skills acquired in the course. Measurements at specified time intervals to calculate a rate is from the context of the question. They should also specify other factors that need to be controlled, e.g. temperature. I'm sorry, I don't know if you can see what I've got on the screen. So in order to gain full marks from the mark scheme, they would need to incorporate both information from the context that they're given, as well as information from the practical that they will have carried out. It's not possible to gain full marks on a devised question by merely repeating what the students have said. I'm just checking the checkbox. Question: are details about the apparatus used also required? The things that are specific to this investigation would be, so students would be required to say that they would use a microscope and graticule to measure the length of the pollen tube, because that is part of understanding this particular investigation. They would not be required to state what apparatus they used to measure out the sucrose solutions or the measure out the boric acid. Standard lab equipment that you would use in anything doesn't need to be specified, but anything that is specific to this investigation does need to be specified. Has anybody, I'll give you a couple of minutes if you've got any more questions about practical investigations and practical questions on exam papers, if you would like to type them in, because it is part of quite an important part of the course. There is no longer any sort of assessment of practicals as they are carried out. All practical assessment is based on questions that will be set in Unit 3 and Unit 6. And for the international A level, there is no, there'll be no practical questions on either on Unit 1, 2, 4 or 5 exams. They will all be on Unit 3 and Unit 6. Right, in Unit 3, the exam will be based on the core practicals in Unit 1 and 2. So Unit 3 will only look at practicals from Unit 1 and 2. You may get asked about, you're very likely to get asked about a practical in a different context. So it will be a practical that's been carried out, but it will be put into a different context. it is very unlikely that students will be asked to just repeat a practical they have carried out. So they need to practice putting them into a different context. Unit 6 can use practicals from any part of the course. Unit 3, that is only units 1, practicals in unit 1 and 2. Unit 6 is practicals from any part of the course. And there is very likely to be a question on devising an experiment, which will be a high tariff question, but there's also questions on parts of practicals. So you might be given a context and asked what factors need to be controlled. Or given some apparatus and ask about what you would measure. And as I've just said, unit 3 is based on units 1 and 2. Unit 6 is based on all of the units. Right, we will move on from practical skills. However, if you've got any questions at the end, there will be time to ask about them later. So we're going to move on now to consider the mathematical skills that are required. And on each one of the exam papers, or on units 1, 2, 4, and 5, at least 10% of the marks available are for level 2 maths. So if you look at Appendix 6 in the specification, or you go back and look at Appendix 6 afterwards, that lists the mathematical skills that are required. It gives examples, but the assessment of mathematical skills is not limited to the examples provided. They can be put into any appropriate biological context. Now, when you look at the mathematical skills, some of them are in bold. The ones that are in bold will only be tested in the IA2 year. So the ones that are in bold will only be tested in units 4 and 5. It's the only part of the specification where you've got this distinction by putting things in bold. So if we have a look at this question, one of the practicals is looking at sugar content. This table shows the concentration of sugars in 3 pineapples. So we're looking at fructose, glucose, and sucrose in 3 different pineapples. And the students are asked to calculate the mean concentration of glucose in these pineapples and to give the answer in grams per decimeter cubed. So which mathematical skills do you think this question is assessing? If you've got the specification accessible, if you look at page 62, that's where the mathematical skills are. But there are 2 mathematical skills that are being assessed in this question. So if you put into the chat box what you think they are. Right, I've got a question about worksheets And I've had other questions about the resources that are available in line with this qualification. Pearson do produce a number of resources that are designed to help with lesson planning, teaching the specification, and exam practice. At the end of this presentation, we will be talking about the support available from Pearson. So hopefully, when we get there, that will help answer that question. If not, then ask me the question again in the chat box. Yes, we are getting some suggestions here, calculating the average and unit conversion. Addition and division is not really a level 2 math skill. When this paper was being written, or this question was being written, as papers are written, there's a record kept of the math skills that are tested to make sure there's 10%. Addition and division wouldn't count as a math skill that contributed towards that 10%. But you're all getting the idea of unit conversion and calculating a mean. And this is 2 of the math skills that are listed in the appendix. And these are the 2 math skills that would have been tested in this question. Recognizing and making use of appropriate units in calculations, and candidates may be tested in their ability to convert between units, and finding arithmetic means. The students were asked, the information given in the table was in grams per centimetre cube. They are asked to give their answer in grams per decimetre cube. So they are specifically being asked to make a conversion. If the mark is being given for the conversion, it will be a specific part of the question. There are many instances where different units that mean the same thing can be used. If it's not specified, then they can use whichever. They can either continue with the units that are given, or they can make a conversion, as long as the conversion is correct, of course. But this specifically asked them to give their answer in grams per decimetre cubed, so they're being tested on their ability to convert between units and to find a mean. And there will be many occasions where they need to find a mean when they're looking at experimental data. Now, even students with stronger mathematical skills don't always apply them in a biological situation. In exam papers, very often, the maths questions are left unanswered when they're really quite straightforward. So please encourage your students to not look at a maths question and think, I can't do this. Marks are lost when they are really quite straightforward calculations. Also encourage your students to show their working. If they get the correct answer, they'll get full marks. But if they don't get the correct answer, there may be a mark available for one of the stages, which they may have got correct. So, which mathematical skills could you practice using this data? You've got a table of data, it's got the time, it's got the concentration of ammonia that's produced by urease, and the concentration of ammonia was recorded every 5 minutes for 35 minutes at a pH of 6. So type into the chat box what mathematical skills you think you could be testing. Are we getting any? These are a few ideas that have been given that could be tested using this data. So we've got converting data, converting it into standard form, plotting the data, calculating a rate of change, plotting the data and finding the rate of reaction at a particular point in time by drawing a tangent to the curve. One set of data, at least 6 different mathematical skills that can be developed using this data. So to help your students develop their mathematical skills, it's worth giving them short exercises as part of a lesson or as homework. Probably not to do all of these things at the same time, but you could give them this data and tell them to go home and convert it to standard form. Then the next time you could tell them to plot the data and calculate a rate of change from the graph. Sets of data like this are very useful for helping develop practical skills, just as part of a lesson, not as a whole lesson usually. Or using their own experimental data, particularly if you've got large enough classes to be able to amalgamate data and develop quite a set of it. We're now going to move on to look at the pre-release scientific article. This is a novel aspect of the Pearson International A-Level Biology. It's a short scientific article, approximately 2,000 words long. It's written in language that's accessible to A-level students. And it's assessed as part of the Unit 5 examination, but the content of the article and its assessment can relate to any part of the specification. It's a synoptic aspect of the assessment. It brings together all aspects of the specification. It's not a simple comprehension exercise. They will make use of the information in the article, but the questions will relate to the science that the students need in order to understand the article. It's released in advance. You will have the opportunity to send your students away to read it and then to go through it with them and talk about it, think about what might be assessed, and make sure that they are very familiar with it. And we're now going to move on from that to think about the command words. And Appendix 7 of the specification lists the command words used in the question papers. Part 2 of this training, which will be in 2 weeks' time, considers this taxonomy in more detail. But it's very important that your students, and therefore yourselves, are familiar with the command words used and what they mean. One of the main reasons candidates underperform is because they don't answer the question that is asked. And this is often because of confusion about the command words used in the question. Students need to be familiar with the command words if they are to understand what they are being asked to do in exam questions. So it's quite, when you're looking at past questions, when you're talking to them about past questions, the command word is as important as the content of the question. So now we've talked about the specification in quite a bit of detail. We're going to move on to how to plan to deliver the content. Now this gives some of the additional guidance that's available from Pearson. Practical skill guides, mathematical skill guides, and topic guides. You've got to, when you're planning how to deliver the content, there is quite a lot to deliver. You've got to make sure that you cover everything in the time you have available. Clearly, the time you devote to teaching each particular topic or specification point will depend on many factors, including the time you have available, the academic ability of your students, and the prior learning and attainment of your students. But science A-level specifications are substantial documents, and the key to making sure you complete the course is planning. As well as the specification, Pearson also produced resources such as the year planner, which help to give you an overview of how the course will be organised. So you need to think about what sort of things you would include in your course planner. To make sure you cover all the content, you've got the specification, you've got the course planner. From the course planner, you've got schemes of work, and then from the schemes of work, you have got your lesson plans. Your year planner should include dates for teaching each topic, dates when you will be revising, and dates when the students will be assessed. You might also want to include some other key dates, such as student review dates and practical dates. It can be helpful to have a clear idea of when practical work is planned, especially if significant preparation is required. And also, some of the practical work is better carried out at specific times of the year, particularly ecological studies. So you need to take that into account. So those are the sort of things you might want to include in your course planner. Now, Pearson do produce schemes of work. And this is an example of a scheme of work produced by Pearson, or part of a scheme of work produced by Pearson. It's from topic 1. It gives the aims and learning outcomes. It gives examples of what you could teach. And then in the 3rd column, it lists the transferable skills that can be introduced when you're teaching this particular unit. It's not a definitive document. You don't need to teach the course as detailed in the scheme of work. A scheme of work is a very personal document, and you're almost certainly to want to deliver some aspects of the course differently. However, it provides a starting point from which you can develop your own personalised scheme. The scheme of work that Pearson has produced is organised in weeks, 34 in the first year, the IAS year, and 33 weeks of teaching in the IA2 year. And for each week, a topic area with aims and objectives is identified. So this is an example of a week's teaching. And then it's got ideas that might help you deliver the topic, classroom activities, teaching resources, and then, as I said, the transferable skills. When you've got your scheme of work, you then need specific lesson plans for each lesson. So this content here might be delivered in 2 or 3 lessons. So if that is your week's work, your week's part of the course that you intend to cover, it's got 3 specification topics, or 3 specification points. Put into the chat box if you're going to say this is maybe 3 lessons, choose one of them and put into the chat box what you would incorporate into your lesson plan. Doesn't have to, it's general things that you would put into a lesson plan. While you're typing that in, I will just answer the question about mathematical qualifications from Maya. If they are specifically in the specification, they are biological mathematical skills, and your students do need to be able to do them, such as Hardy-Weinberg calculation, diversity calculations, standard deviation. Standard deviation would be in the second part in IA2. But yes, they do need to be aware of how to carry out those calculations. They would be given the formula, but they need to know what information they need to put into that formula in order to carry out the calculation. And past papers will have examples of questions using Hardy-Weinberg or using standard deviation. So yes, if it's part of the specification, if it's a topic point, they would be expected to know how to carry it out. If we could have a few ideas about lesson plans now, that'd be helpful. Question about the course planners. They are available on Pearson's website. When we get to the final part of this presentation, it is about all the support that's available from Pearson, and it does say where you can find the resources that are available. There's quite a number of resources. And then we move on to we've got quite a lot of ideas of what could be included in lesson plans. So what you might want to include in any lesson plan is the key concepts of what you hope your students will learn in this lesson, the sequence of events that you're going to go through, the resources required, and then it is very important that you finish the lesson with some aspect that will demonstrate that learning has taken place. You might also want to identify opportunities to develop mathematical skills, practical skills, and transferable skills. But like a scheme of work, a lesson plan is something personal to you, and it should be something that helps you deliver your teaching. Lesson plans and schemes of work should be evolving documents. So when you get an opportunity, it's useful to reflect on how up-to-date and useful your lesson plans and scheme of work are. Now, when it comes to planning practical activities, many students find the acquisition of practical skills challenging. One core practical is to investigate the vitamin C content of food and drink. This core practical could be used to teach practical skills. You could use it to develop aspects of planning, implementation and measurement, or processing results. So you might want students to carry out the whole practical. You might want to use it to help them devise an investigation, in which case you would probably give them a specific food or a number of different foods. You might want to help them develop implementation and measurement skills or processing results. So if we move on, these are some, you don't have to develop all the skills in one practical activity. You can break it down to help so your students develop different aspects of practical work in different types of lessons. So you might provide a method, but ask students to devise their own investigation. You might ask students how you can make their data more reliable, and hope they would come up with the suggestion of repeating titrations and calculating mean values and the use of error bars. You might want to ask students to evaluate methodology by providing different methods and asking them to discuss the advantages and disadvantages of each. For instance, when you're measuring vitamin C content, you might do it by counting the drops of DC-PEP, or you could make it more accurate by using a burette to titrate the DC-PEP. Now we need to look at the assessment design. So as we said, there are 6 external examinations. This just shows you the 6 exam papers, 1, 2, and 3 for AS, 1, 2, and a further 1, 2, and 3, 4, 5, and 6 for the A2. They're all external examinations. It's expected that the IAS topics would be covered in the first year and the IA2 in the second year. All assessments are compulsory and there are no optional units. So if we have a look at the Unit 1 and Unit 2 examinations, they are both 1 hour 30 minutes long. They are both available in January, June, and October. They both carry 80 marks. And that 80 marks is 40% of the IAS, or it contributes 20% of the IAL. This equates to approximately 1 minute for each mark. There's no synoptic assessment in these units. So in actual fact, candidates could take the Unit 2 examination after studying topics 3 and 4, before studying topics 1 and 2, although this is not recommended because it is designed to develop biological knowledge. So you've got an overview of the content there, transport and health and membranes, proteins and DNA in Unit 1, cell structure, reproduction and development, and plant structure and function, biodiversity and conservation in Unit 2. In the exam papers, the assessment will consist of a combination of multiple choice, short, open response, calculations, and extended writing questions. There will be points-based and level-based marks. There'll be a minimum of 8 marks targeting maths at level 2 in each paper. And there will be application of knowledge and understanding to both familiar and unfamiliar contents. All papers will have 10% of maths marks at level 2. So because it's modular, when you come to cash it in, not all candidates will be using the same combination of papers. The overall qualification has to have 10% of marks for maths, so each paper is designed that way. If we come on to look at Unit 3, again, this is a written examination. It lasts for 1 hour, 20 minutes. It's available in January, June, and October. It has 50 marks, which makes 20% of the IAS and 10% of the IAL. Now, as it's worth 50 marks, that is approximately 1.5 minutes per mark. It examines the practical skills acquired studying topics 1 to 4. There's no multiple choice or level-based questions in this exam. There's short open response and calculations questions. There's points-based and level-based marks. So a minimum of 5 marks targeting maths, because it's 10%, and it's application of knowledge and understanding to familiar and unfamiliar context. If we move on to look at Unit 4, sorry, I was just checking on the chat box again. This shows the key elements for Unit 4 and Unit 5. They're both written exams of 1 hour, 45 minutes. available again in January, June, and October. They carry 90 marks. They form. That is incorrect. They do not form any of the IAS. So ignore that, but they form 20% of the IAL. And Unit 4 covers energy flow, ecosystems and the environment, and microbiology, immunity and forensics. Unit 5 covers respiration, muscles and the environment, and coordination, response and gene technology. And again, they have multiple choice, short open response calculations and extended writing questions. There are point-based and level-based marks. 10 to 15 marks on each paper draw on IAS topics. So these papers include material learned for the IAS exam, so they are synoptic. There's a minimum of 9 marks, again 10%, targeting maths at level 2. Application of knowledge and understanding to familiar and unfamiliar contents. And Unit 5 includes the pre-release scientific article. Which could be based on any aspect of the IAL course. So that is any of the 4 knowledge-based units. Unit 6 completes the IA2 assessments. It's similar to Unit 3, 80 minutes, worth 50 marks, which is approximately 1.5 minutes per mark. And it assesses the practical skills acquired throughout the whole course, topics 1 to 8. And there's no multiple choice in this. And again, there's a mistake here, I think, because it's not part of the IAS. So it's not 20% of the IAS. We're going to briefly now look at how grade boundaries are set. The raw mark on a paper is used to calculate a final mark, or a UMS, unified mark, for the unit. Candidates are then given a grade for each unit. The final grade for the qualification is determined by the aggregated UMS marks for all the units. And the link takes you to a calculator that allows you to convert raw marks into UMS, although these are different for each of the units. And we're going to move on to look at assessment objectives. When the exam papers are being prepared, there are 3 assessment objectives that are covered throughout the exam throughout the exam paper. And these identify the way in which biological knowledge and understanding will be assessed. The AO1 questions, demonstrate knowledge and understanding, are the straightforward questions assessing the learning of biology detailed in the specification. For instance, if candidates were asked to explain how primary structure determines the shape of a protein, this is something that comes straight out of the specification, so it is an AO1 question. AO1 questions will account for approximately 35% of the total marks available. AO2 is application of knowledge and understanding of science in familiar and unfamiliar contents. So if candidates were asked to explain the role of the primary structure of the calcium channel protein in its function as the channel protein, candidates would need to think about what calcium ion channels do, where they are found, and link the folding and location to particular places on the protein. This is not explicitly stated on the specification, So this would be AO2 and AO2A. And AO2A, again, is approximately 35% of the marks in the IAL qualification. AO2B is analysis and evaluation of scientific information to make judgments and reach conclusions. So questions that ask candidates to interpret data are assessing AO2B. And this will be approximately 12 to 13% of the marks across all the examinations. AO3 is specifically experimental skills. That will account for about 16% of the marks, and any question assessing practical skills is assessing AO3 and will be found only in the Unit 3 and Unit 6 examinations. Now, when the paper is produced, it also has a mark scheme. And as I said earlier, this is developed having had a look at students' responses. But the purpose of a mark scheme, it's written at the same time as the paper itself. It's revised in the light of student responses, and it's used by all the examiners to mark student responses. It's to ensure that candidates are rewarded for correct, relevant biology, and that examiners mark consistently across all candidates. After the exam, the mark schemes are published and can be a useful resource to teachers and students. The mark scheme is written for a particular paper and is revised at pre-standardisation prior to its application. Mark schemes for AO1 questions should be reasonably consistent for the duration of the qualification. For many AO2A, AO2B and AO3 questions, although the information being tested is the same, the context may well change between subsequent exam papers. So this will change from one paper to the next, so the mark scheme may change significantly because of the different context in which the mark questions are set. So if we move on to a question, sperm cells contain mitochondria. Describe the function of mitochondria in the movement of sperm cells. Now that's 2 marks, so you would have 2 minutes to write an answer that gain these 2 marks. So jot down on a piece of paper what you would write as the answer to this question, and then we will go on and look at the mark scheme. Right, the mark scheme for this question describe the function of the mitochondria. So this is what the mark scheme would look like. So the mitochondria carry out aerobic respiration to provide ATP, which is required to move the flagellum. 2 marks for the question, 3 marks available. Very often there are more marks available than are required to get full marks, but obviously you can only get 2 marks. You get a mark, each statement carries a separate mark. The first column gives the preferred response. The additional guidance helps examiners interpret the answers candidates produce. So we would accept respiration rather than aerobic respiration, but we would not accept produce or make energy. It has to be provide. Now, so we then think about which specification point and assessment objective it is looking at. Hang on, I missed a couple of pages. No, I haven't. Sorry. So the specification point that this was addressing was understand how mammalian gametes are specialised for their functions and understand the role of the mitochondria. And it was an AO2A because it was application of knowledge and understanding of science in familiar and unfamiliar contexts, because it was using the mitochondria and putting it into the context of mammalian gametes and asking them to apply it to that context. We're now going to move on to actually look at the exam paper. The front cover provides paper details at the top and space for the candidates to enter their details. I would encourage your students to get into the habit of checking they've been given the correct paper, as well as filling in the correct candidate and centre numbers. The other information, equipment they will need, time, they should be very familiar with this, but it's still worth checking. Then we get the instructions on the exam paper and the information. Again, you should have made them very familiar with this, but it is still worth getting them to check it. Incomplete examination papers suggest that many candidates run out of time and that the majority start on question 1 and work through sequentially to the last question. Now, you need to think about the advice you would give to your students to spend on each question and what order you would recommend that teaching that the questions are answered in. There's no real right and wrong answer to the order. Some students might prefer to start with ones that they're more familiar with. Most just work through the paper because of the way it's set out. You should advise your students to make sure that they leave time to answer the higher tariff questions. And again, remind them, it's about a minute a mark. So if they're spending too long thinking about a question, they should move on to the next one. Teaching good exam technique is as important as teaching the content of the biology syllabus. Students often let themselves down with poor exam technique. Practice using a whole paper and understand how it's laid out. Encourage your students to read the question carefully, paying particular attention to the context and to the command word. If it's a higher tariff question, encourage them to plan their answers. And we always provide more than enough paper. You don't need to fill the whole booklet. Now, a walking, talking mock is a good way to help students develop their exam technique. To make them really familiar, it's best if they sit in the same exam room where they will do their exam, preferably in the same seats, and use an authentic examination booklet, a photocopy of a past paper. They should experience using the examination booklet, not reading questions and then answering on loose sheets of paper. Now, the most important part of this is to talk students through the paper. Go through the question paper, question by question, explain how much time they should spend on each question. You might suggest color coding questions according to time allocation, draw their attention to context and suggest ways of highlighting keywords. Higher tariff questions, explain how they may plan an answer. Explain there are different ways to approach an examination paper. And then when you've gone through it with them all, they should be allowed to write their responses in timed conditions, giving them the same length of time as they would have for the exam. When they're finished, you might want to provide the students with a copy of the mark scheme and maybe the examiner's report and get them to peer mark each other's papers. After peer marking, you might find it useful to have a discussion about some of the points that were contentious. And you will probably need to provide some professional input to guide students as why a mark can or can't be given. And then at the end, you might want to ask your students to fill out a table to say if they felt less the same or more confident before or after the mark. We're going to finish off this session by looking at the support that is available for Pearson, from Pearson. I have just read the comment, which I'll quickly address, about the mark scheme saying that the difference between providing energy and producing energy. It is a concept students find difficult, but they should have been taught from the age of about 11 that energy cannot be made or destroyed. It can only be converted from one form to another. And it is an extension of this that means that we do not accept production of energy, because energy can't be produced. It's a fundamental concept of science, and it is something that students just, I appreciate they find it difficult, but it's something they just have to accept. The level 2 math skills are listed in the appendix. So if you look at what's there, that will tell you that they're all level 2 and they're the ones that will be tested, the ones that are listed. So we're going to finish off with looking at the support that's available from Pearson. This is a list of what's available. For planning, teaching and learning, we've got the Getting Started Guide, the training, schemes of work, skills mapping, SAMs, examiner's reports. Then we've got things to help with examination, preparation and assessment. And then there is support after the results come out. A number of materials are available on the website. Now, a number of you have been asking about this. If you are teaching the Pearson A-level qualification, that will give you access to the materials available on the website. And the web page has got illustrated, has got 4 tabs. It's got specification and sample assessment materials, exam materials, teaching and learning materials, and forms and administration. The teaching and learning materials is where you will find lesson plans, course planner, more exemplars, and much more. And this bank of materials is continually being added to. Results Plus is a tool that teachers will find useful in analysing their students' exam results. It's a free online results analysis. It provides a detailed breakdown of student performance. So this identifies topics and questions where students could benefit from further learning and inform teaching strategies and approaches. It benchmarks your school's performance against other Edexcel schools. And it's not just a post-result service. Mock exam results can also be fed into the system to produce analysis, which will help students and yourselves identify where they need to focus for the exams they are preparing for. And Results Plus Direct gives your students access to their final grades and performance breakdown wherever they are. And it's free for schools to sign up for this. So students take the exam paper. The exam papers are scanned into the system that enables them to be marked. Using ePen, the examiners mark the papers online, which means that the papers are marked by different people, and then the performance reports are shared. There is also Exam Wizard, a tool that teachers may find useful in producing exercises and past papers for students to use for study or in producing tests. Uses questions from past papers and the mark schemes that were used to mark these papers. And you can use it to produce bespoke questions, tests on particularly just on one topic or several topics. It is a very useful resource. In addition to that, Access to Scripts is a free online portal which allows teachers to immediately access electronically marked papers. It's self-service, online, free, and instant from results day onwards. It cuts down on speculative EARs, as students have the time and information needed to make informed decisions. You can see all of your students' marked exam papers free of charge. It's accessed through Edexcel online. The scripts don't have any examiner or annotation on them. They do have the mark that was awarded for the question, but no examiner annotation. And you can access it alongside Results Plus, another free online service. Pearson Publishing produces a modular, it supports a modular approach. These books are produced to be in line with the latest specifications, which are recognised by universities worldwide. They've got exam practice in them, gives a real world context, and they're reviewed by a language specialist to ensure the book is clear and accessible. Now, you have a dedicated subject advisor, which is Tim Lawrence, who will help you with any questions that you have that have not been answered this morning. He's easy to get hold of. If he cannot get back to you immediately, he will respond as soon as you can, and you can contact him via the information on the slides. Any questions that I didn't fully cover about access to any resources, course planning, lesson plans, things like that, worksheets, if you contact Tim, he will be able to help you access the resources available. All the exams are paper-based. There's no on-screen exams available at the moment. At some point in the future there might be, but it's paper-based exam. Students writing the exam booklet. These all get sent away, and then they get scanned into the ePen system, which means that they are marked online, but the questions are not actually answered online. Whether you should discard a book is debatable, but it has been superseded by this book, which goes with the book goes with the course as it was updated. Can you, if you contact your subject advisor, he will be able to tell you when the pre-release material will be available. It's always helpful if students read about the topics they are studying, and scientific papers can be helpful, but they need to be accessible. They won't benefit from reading papers that they don't understand. And students are not necessarily very good at identifying the words that they need to understand and the words that they don't need to understand. Well, that brings the presentation to an end. Is there any more questions that anybody has? Again, I'm not 100% sure if the PowerPoint presentation is shared afterwards, but your subject advisor will be able to help you with that. I think it's 20%. I think it's 20 marks for the scientific article, but if you look at a past paper or ask your subject advisor, that can be confirmed. The command word describe and explain. The second part of this training looks in more detail at assessment and at student responses and at mark schemes. So we will cover it in much more detail there. But describe is something that is taken directly from the specification. Explain is applying what's in the specification to a slightly new context. The experiments are listed in the practical, in the context of the units, the ones that are core practicals. Right, that brings the presentation to an end. If there's any more questions, right, thank you everyone for your attendance this morning. I hope this presentation was useful to you, and hopefully I will be talking to most of you in 2 weeks' time for the second part. Module 2 is 2 weeks today on the 29th at the same time.
Jill, there's a question in regards to the book module on how they can book it.
Yeah, I don't know how. I assume that module 2 is booked in the same way as this one. Ah, yes, someone's confirmed that. use the same link as for this one.
May I know what is the purpose of think bigger, you know, in the textbook?
Sorry, say that again.
I mean, think bigger topics, you know, after each section of the, you know, after each chapter, you know, we can see that topics think bigger, you know. What is the purpose of that?
Thing it is just to engender students' interest in biology and to see how what they're learning fits into the big picture. But in terms of, I mean, when you're teaching biology, you want your students to come away with an interest and knowledge of biology. You also want them to pass their exams. The specification will list exactly what will be on the exams. Wouldn't like to think you would only teach to exams, but if you focus on the points in the specification, they are what will be in the exams. Does that help?
Thank you.
Thank you, everybody. I think that brings this meeting to an end. Is there anything else, Nasaka?
There's nothing else at the moment. I think just there's one last comment in regards to sharing a link to module 2 webinar. I think they will send it out, I believe.
I had no input into actually signing up for it. So it would require somebody else to send the link out.
Yeah, I believe Pearson would send the link out
Yeah.
before it's due to attend. So the
Yeah.
link will be sent. Yeah, I think that's it. I'll end the recording if that's all right. You're welcome.